Our next presenter is Ting Wang from Southeast University. And she's going to be telling us about genetically informed brain morphometric similarities revealing suicide risk in bipolar disorder.
Hello everyone, my name is Wang Ting from Southeast University in China. The research I'm presenting today is genetically informed dissociative brain morphometric similarities revealing suicide risk in bipolar disorder. Suicide with pronounced heritability, especially in psychiatric disorders such as bipolar disorder, has caused major social burdens on individuals and countries. And the genetic architecture of suicide assumes a host of independent allelic variation, each of small evident, contributing to the tendency of kill oneself. Individuals with the greatest accumulation of genetic predisposition have higher suicide tendency. To date, the genetic risk for suicide has been revealed more clearly through a series of GWAS and post-mortem human brain transcription studies. Accumulating evidence has also been captured to characterize suicide as a dysconnectivity syndrome, including selective change in brain structure. However, multifaceted genetic landscape, including genetic variation, translational abnormalities, and downstream maladaptive changes, biological process responsible for the anatomic alterations with suicide effect is currently underexplored. Morphometric similarity network map, including surface area, cortical thickness, gray matter volume, Gaussian curvature, mean curvature could capture more brain connections within the same cytoarchitecture class. This has been validated in primate brain. Cumulatively, growing neuroimaging genetic studies utilize the translational mobility model to spatially associate the cortical expression map with the cortical altered MSN map. In psychiatric disorders, including the bipolar disorder, major depressive disorder, they attempt to identify the normative gene expression gradients enriched for disease-specific genes accountable for the corresponding MSN chains to further review the hidden pathophysiology mechanism. So, the genetic variations, including transcriptomic factors that drive replicable suicide-relevant morphometric alterations in bipolar disorder are still poorly understood. So it highlighted a gap in our understanding of genetic landscape of suicide risk in bipolar disorder. Moreover, the convincing proof of principle with paired measures of multi-biological characteristics is also urgently needed. In this study, we want to decode reproducible anatomic definite suicide effects informed by morphometric similarity organization using multi-biological characteristics into independent bipolar disorder cohorts, composed patients with or without suicide attempt, and test it on the genomic axis. All the procedures contributed to this work complied with the ethical standards of the relevant national and institute community on human exploitation. In the current cross-sectional study, the discovery cohort comprised 135 bipolar disorder patients and 96 health controls recruited from the affiliated brain hospital from Nanjing Medical University, and the replication cohort contained 46 bipolar disorder patients and 23 health controls from another hospital. This is our study overview. We genetically decoded the anatomic definition of suicide effects, informed by the morphometric similarity organization use multiple biological characteristics in their discovery and replication cohorts. First, we morphometrically evaluated MOSM attenuations with suicide effects and query the transcriptomic correlates and dominant neurotransmitter receptors responsible for the MOSM attenuations with suicide effect. And next, we molecularly validated the MOSM deafness by suicide effects by paired measures of genotype data from target SNP sequencing. The last, the validation of the brain morphometric and related transcriptional patterns from an independent replication cohort was also did. Firstly, the morphometrically informed MSM attenuations with suicide effects. Regional MSM differences between BDSA and BDNSA groups were found in interrenal cortex and left lateral occipital part 7, and by transcriptomic correlates of altered MSNs with suicide effects. The neurotransmitter receptor was also indicated by the enrichment analysis. Whereafter the morphometrically informed deafness by suicide effects in chemo architectures was decoded by multiple linear regression model combining nine neurotransmitter systems significantly. Opioid receptor was identified for its most contribution to the above significant fit of the model in the dominance analysis. And by targeted sequencing, BDSA showed a significant higher MGPS of OPRM1, which including the mu opioid receptor indicated by the dominance analysis compared with BDNSAs. Consistently, morphometric profiles in interrenal cortex and left lateral occipital part 7 demonstrated their differences between BDSA and BDNSAs. Their dissociative morphometric profiles all displayed obvious correlations with MGPS of OPRM1. Accordingly, we tested whether these regional morphometric profiles mediated the effect of OPRM1 genetic risks on suicide attempted using multi-level mediation analysis. The effect of its genetic risks on extendency of suicide attempted was fully positively mediated by the regional MSM of interregional cortexes. And in an independent replication cohort, morphometrically assay-related MSM chains were spatially correlated with the discovery cohort. Left in regional cortex was also showed obvious differences in MSM between BDSA and BDNSAs. For the related transcriptional patterns, it suggested being remained largely the same, either the overlapped genes or pathways, or the enrichment network, especially for motion morphe addition from previous Java studies. Consequently, abnormal cytoarchitecture communities, especially maladaptive chains in neurocommunications between the interrenal context and the reward circuit regulated by opioid receptors reflected by the enhanced morphometric similarities could mediate the effect on the increased suicide tendencies involved OPRM1 gene variants in bipolar disorder. And some limitations I want to highlight. Firstly, our sample size is small, and large sample size is needed to further validate our findings in the future. And the gene expression data was obtained from the post-mortem brains of 6 non-Chinese donors, but the structure MRI data was obtained from the living brains of 300 Chinese participants. The inconsistency between the transcriptomics and the brain image data could reduce the statistic power of identifying the correlating genes with small effects, and the interpretation remained influential given the structured nature of the data and required confirmation. What we are doing is through longitudinal studies to tracking the network changes over the time. And finally, I would like to thank my team and thanks for listening.
So thank you very much also for this very nice presentation. There are no questions yet from the public. Okay, so I've got one. The opioid receptor densities. I must have missed this here. You said that there were differences between in opioid receptor densities between entorhinal cortex and the areas of the reward system.
I get the opioid receptor from the multiple linear regression by combining 9 neurotransmitter systems, and the opioid contributed the most contributions. So we by targeted sequencing, and we found the MGPS of OPRM1, which included the opioid receptor. The MGPS of OPRM1 in BDSA and BDNSs make differences. So we make the multi-level mediation analysis, and we found it have a significant results.
Right. And when you say serotonin, you really mean the neurotransmitter serotonin or serotonin receptor?
Sorry, my English is not well.
Yeah, and don't worry, my Chinese is worse than your. I mean, so my Chinese is 2 words, so don't worry. Thank you. That's not a problem. So it's this. What do you know? What marker was used to visualize the serotonin receptor?
We targeted sector 7 variants, but I didn't show it in my slides.
Oh, okay. Well, then I missed that. I'll have to read the article. It was just getting too late in the evening for me to get this. I'm sorry.
I'm sorry.
Thank you very much. All right. Is our last speaker here tonight? Is Jack Dodson with us? Yes, it looks like it. No? No? Okay. I think that's the end then. Thank you very much for following this session, and see you tomorrow.
But before you go, sorry, we've supposed to remind you to rate the sessions, please, and to do it like immediately after the session or as quickly as possible, because otherwise we forget to do it, like if we don't do it immediately. And it helps the organizers, the speakers and everything to know, okay? So use the app to rate the session. Thank you.